Section number 6 of Gray's Anatomy Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Spinal Cord Part 2. Structure of the gray substance. The gray substance consists of numerous nerve cells and nerve fibers held together by neuroglia. Throughout the greater part of the gray substance, the neuroglia presents the appearance of a sponge-like network, but around the central canal and on the apices of the posterior columns, it consists of the gelatinous substance already referred to. The nerve cells are multipolar and vary greatly in size and shape. They consist of 1. Motor cells of large size, which are situated in the anterior horn, and are especially numerous in the cervical and lumbar enlargements. The axons of most of these cells pass out from the anterior nerve roots, but before leaving the white substance, they frequently give off collaterals, which re-enter and ramify in the gray substance. 2. Cells of small or medium size, whose axons pass into the white matter, where some pursue an ascending and others a descending course, but most of them divide in a T-shaped manner into descending and ascending processes. They give off collaterals which enter and ramify into the gray substance, and the terminations of the axons behave in a similar manner. The lengths of these axons vary greatly. Some are short and pass only between the joining spinal segments, while others are longer and connect more distant segments. These cells and their processes constitute a series of association or intersegmental neurons, which link together the different parts of the medulla spinalis. The axons of most of these cells are confined to the side of the medulla spinalis in which the nerve cells are situated, but some cross to the opposite side through the anterior commissures and are termed crossed commissural fibers. Some of these latter end directly in the gray substance, while others enter the white substance and ascend or descend in it for varying distances, before finally terminating in the gray substance. 3. Cells of the type 2 of Golgi, limited for the most part to the posterior column, are found also in the substantia gelatinosa of Rolando. Their axons are short and entirely confined to the gray substance, in which they break up into numerous fine filaments. Most of the nerve cells are arranged in longitudinal columns and appear as groups on transverse section. Nerve cells in the anterior column. The nerve cells in the anterior column are arranged in columns of varying length. The longest occupies the medial part of the anterior column and is named the anteromedial column. It is well marked in C4, C5, again from C8 to L4. It disappears in L5 and S1, but is well marked in S2, S3, and S4. Open parens, Bruce, close parens, see footnote. Footnote, Topical Atlas of the Spinal Cord, 1901. End footnote. Behind it is a dorsomedial column of small cells which is not represented in L5, S1, S2, nor below S4. Its axons probably pass into the dorsal rami of the spinal nerves to supply the dorsal musculature of the spinal column. In the cervical and lumbar enlargements, where the anterior column is expanded in a lateral direction, the following additional columns are present, that is, A. Anterolateral, which consists of two groups, one in C4, C5, C6, the other in C6, C7, C8 in the cervical enlargement, and of a group from L2 to S2 in the lumbosacral enlargement. B. Posterolateral in the lower 5 cervical, lower 4 lumbar, and upper 3 sacral segments. C. Post-posterolateral in the last cervical, first thoracic, and upper 3 sacral segments, and D. Acentral in the lower 4 lumbar and upper 2 sacral segments. These cell groups are evidently related to the nerve roots of the brachial and sacral plexuses and supply fibers to the muscles of the arm and leg. Throughout the base of the anterior column are scattered solitary cells, the axons of some of which form cross-commissural fibers, while others constitute the motor fibers of the posterior nerve roots. Footnote. Lenhasek and Cahal found that in the chick embryo, the axons of a few of these nerve cells passed backwards through the posterior column and emerged as the motor fibers of the posterior nerve roots. These fibers are said to control the peristaltic movement of the intestine. Their presence in man has not yet been determined. End footnote. Nerve cells in the lateral column. These form a column which is best marked where the lateral gray column is differentiated, that is, in the thoracic region. But it can be traced throughout the entire length of the medulla spinalis in the form of groups of small cells which are situated in the anterior part of the formatio reticularis. In the upper part of the cervical region and lower part of the medulla oblongata, as well as in the third and fourth sacral segments, this column is again differentiated. In the medulla, it is known as the lateral nucleus. The cells of this column are fusiform or star-shaped and of a medium size. The axons of some of them pass into the anterior nerve roots by which they are carried to the sympathetic nerve. They constitute the white rami and are sympathetic or visceral efferent fibers. They are also known as preganglionic fibers of the sympathetic system. The axons of others pass into the anterior and lateral funiculi, where they become longitudinal. Nerve cells in the posterior column. 1. The dorsal nucleus. Nucleus dorsalis, column of Clark. Occupies the medial part of the base of the posterior column and appears on the transverse section as a well-defined oval area. It begins below at the level of the second or third lumbar nerve and reaches its maximum size opposite the 12th thoracic nerve. Above the level of the 9th thoracic nerve, its size diminishes and the column ends opposite the last cervical or first thoracic nerve. It represented, however, in the other regions by scattered cells which become aggregated to form a cervical nucleus opposite the third cervical nerve and a sacral nucleus in the middle and lower part of the sacral region. Its cells are of medium size and of an oval or piriform shape. Their axons pass into the peripheral part of the lateral funiculus of the same side and there ascend, probably in the dorsospinocerebellar, direct cerebellar, fasciculus. 2. The nerve cells in the substantia gelatinosa of Rolando are arranged in 3 zones: a posterior or marginal of large angular or fusiform cells, an intermediate of small fusiform cells, and an anterior of star-shaped cells. The axons of these cells pass into the lateral and posterior funiculi and there assume a vertical course. In the anterior zone, some Golgi cells are found whose short axons ramify in the gray substance. 3. Solitary cells of varying forms and size are scattered throughout the posterior column. Some of these are grouped to form the posterior basal column in the base of the posterior column, lateral to the dorsal nucleus. The posterior basal column is well marked in the gorilla, Waldeyer, but is ill-defined in man. The axons of its cells pass partly to the posterior and lateral funiculi of the same side, and partly through the anterior white commissure to the lateral funiculus of the opposite side. Golgi cells, type 2, located in this region, send axons to the lateral and ventral columns. A few star-shaped or fusiform nerve cells of varying size are found in the substantia gelatinosa centralis. Their axons pass into the lateral funiculus of the same or of the opposite side. The nerve fibers in the gray substance form a dense interlacement of minute fibrils among the nerve cells. This interlacement is formed partly of axons which pass from the cells in the gray substance to enter the white funiculi or nerve roots, partly of the axons of Golgi cells which ramify only in the gray substance, and partly of collaterals from the nerve fibers in the white funiculi which, as already stated, enter the gray substance and ramify within it. White substance, substantia alba. The white substance of the medulla spinalis consists of medullated nerve fibers embedded in a sponge-like network of neuroglia, and is arranged in 3 funiculi: anterior, lateral, and posterior. The anterior funiculus lies between the anterior median fissure and the most lateral of the anterior nerve roots. The lateral funiculus between these nerve roots and the posterior lateral sulcus, and the posterior funiculus between the posterior lateral and the posterior median sulci. The fibers vary greatly in thickness, the smaller being found in the fasciculus gracilis, the tract of Lissauer, and inner part of the lateral funiculus, while the largest are situated